So, ladies and gentlemen, it's my utmost honor to introduce Dr. Bruce Mcnulty. So Bruce is a distinguished professor here at the University of California, Irvine, and the director of the Polaris Brain Dynamics Research Group at the University of Lethbridge in Canada. So he's a hero of the two worlds. And he started his career in neuroscience as a PhD student in psychology in Canada, and he was a disciple of Donald Hebb, whose portrait used to be a prominent fixture in his office. I don't know about now, like this. First thing you see, I'm sorry. So in his book of the organization of behavior, Hebb had two main ideas: synaptic plasticity and the cell assemblies. Those were theoretical intuitions, and Bruce set out to prove them both experimentally. So as a student, he did pioneering work on synaptic plasticity in vivo, then started his hunt for the cell assembly and started looking at hippocampal place cells, first with John O'Keefe. And then he became the hero of the three words because he made contributions to technology, experimental evidence, and theory. Of course, he invented the tetrodes and spike sorting. And this was a big paradigm shift because we went from characterizing single cells one at a time to characterizing information encoded in populations of neurons. So nowadays we have very high-tech probes with 1 billion different contacts, but the basic idea is still what Bruce proposed, and it's an innovation that is the basis for thousands and thousands of publications for all fields in neuroscience. So with this tool, Bruce, together with Carol Barnes, went on to acquire an impressive body of evidence on the dynamics of hippocampal ensembles and place cells, providing the first contemporary view on concepts like place field remapping, population vectors, path integration, and so on. He did pioneering work on sleep replay and proposing it as a mechanism for memory consolidation. And in theory, so Bruce made several advances, path integration and attractor neural networks that inspired my own PhD thesis even before I met him. Introduced to the field of hippocampal research Bayesian information, Bayesian decoding, informational theoretical techniques, and these are things now we couldn't even consider doing without. So his paper from 1995 with Jay McClellan and Randy O'Reilly on dual memory systems and replay is a cornerstone of our theoretical framework to understand memory processes. And I guess we'll hear the evolution of his thinking and his ideas. So I should also say that Bruce has been a tremendously prolific mentor. And I mean, if we look around here, you see so many alumni of the Barnes McNaughton lab everywhere. And many of them are really great scientists. And so we mentees got from Bruce great technological tools, great ideas, and a contagious passion. So I suspect we will hear a very passionate talk from him. So please help me welcome Bruce Mcnulty.
Thanks, Francesco, for that very flattering introduction. Oh, this has been an awesome conference. And I don't know whether there'll be a formal acknowledgement, but before I start my talk, I think we need to give a round of applause to Manuela Iasa for organizing the most fantastically well-run conference that I've been to in a very long time. So. So this conference has been not only very well run, but it's had a cast of really high-power individuals, and the quality of the plenary lectures that have preceded me give me a sense of pause and anxiety because it's a tough act to follow, and there's an even better one to come. So if my voice quakes a little bit, you'll know why. So I better get right into it. These are my sources of past and current funding. And I put this slide up here at the beginning because I want to make a little message to the younger generation of scientists. All of the things that Francesco talked about came about because of my habit of collaborating with others and going and finding excellent people to work with, both at the level of students and faculty collaborators. So the message there is that the more and better collaborations you can have in your career, the more likely you're going to be standing up here in a few years giving plenary lectures. So let's get right into it. As needs no saying that the hippocampus is the highest level of association cortex, Larry Squire actually got it wrong. It's like this. And I think of the hippocampus and the cortex more or less like this. The simpler, the better. The best models are the ones you can write on the back of an envelope at the bar. As long as you don't spill too much beer on it, you can usually make progress with it. And so hippocampus sits at the top of the cortical hierarchy, and these little recycle arrows here are meant to indicate to you that it's not just CA3 of the hippocampus that has auto-associative properties, it's all over the brain. All of the cortical modules, probably, particularly in higher cortex, have this characteristic of acting, of having what we now call attractor dynamics. And that figures heavily into what I'm going to talk about today. And then, just to lead in with the standard model, I don't think the standard model in this crowd needs much explanation. This is what pretty much everybody thought up until a few years ago. And so part of what I'm going to be saying today is the sort of refinement of this model that has played heavily already in much of what's been said at this meeting. And of course, as Francesco said, I was very heavily influenced by Donald Hebb, who was a professor emeritus at Dalhousie when I was there. And when I showed him the first evidence for cooperativity or associativity in LTP, he said, well, that's all very nice, but why are you studying this? It's obvious it has to be true. Why aren't you studying cell assemblies? And that is where my career comes from. So, about 87, Richard Morris and I published a paper where we tried to lay out some of the ideas in more modern form that Hebb had come up with, based largely on my readings of the works of David Marr, who's also going to figure heavily in this talk. And to start off, David Marr laid out 3 constraints on the storage capacity, or the number of events that can be stored in such a recurrent associative network: connectivity, sparsity of coding, and pattern separation. And I'm going to touch on all of these, but first on the idea of connectivity. So if you just go backwards for a second, this is the idea. You see a network of pyramidal cells with recurrent collaterals with modifiable synapses in the dendritic tree, and one stores different patterns of input in that by modifiable synapses there. And the problem is that it won't work. If you do the math and you think about the cortex as a randomly connected neural network, as some of us modelers have a tendency to think about at some levels, it simply can't work. In fact, the connectivity of the cortex is so sparse that if one were to model it and make a simple model, one might neglect to add synapses altogether because there just aren't very many of them. On average, each cell in the cortex receives input from only about 1 in a million other cells. And this fact probably underlies much of the reason for the organization of the hierarchical organization of the cortex with the hippocampus at the top. And David Marr was quite aware of this, and I'm going to quote a little chapter and verse from the Gospel according to David. First of all, at a given moment, the animal's brain has information whose expression is now as sophisticated as the animal can provide. It can further classify the information later on, but at the moment it just needs to store the data, and that's the central problem of the lack of connectivity. And that's expressed here. It's required that this should be recorded in some way so that the firing of a few of the cells which were active together in some event can later elicit the firing of all cells active in the event. And that's what he called pattern completion. And then to hint at what I'm going to be talking about shortly, he also said that the final efferent projection from the memory, the simple memory, which he called the hippocampus, should perform little more than an addressing task. And this is the central idea of this talk. And the idea was elaborated by Tyler and DeSena back in '86, who published a very influential paper, at least for me, called The Index Theory, and more recently a revision of, or a revisitation of the theory by Tyler and Rudy. And basically, in this idea, they simply said that the hippocampus creates some kind of an addressing system that, such that if it were possible to retrieve the address or the index and project that back to the cortex, that would be sufficient to recall the attributes of the experience which are actually stored in the cortex, not in the hippocampus. So that's the key element. This is what they actually said. Ironically, the caveat in this scenario is that no content is stored in the hippocampus. It's merely an addressing or an indexing system. So, to give you a little more modern concept of how this works, consider this randomly connected network. So this is a connectivity matrix showing sparse connectivity. If we were to collect all the connections together in modules of about 10,000 neurons, then if each neuron sends and receives 10,000 synapses, those can be densely connected modules, and each of them can act as a associative memory with high storage capacity. The problem with this idea is that these exhaustively connected modules don't have any connections left over to get together associations in other modules, and so you have to dilute the size of the module or the connectivity and configure it in a way that there's something left over for long-range connections. And so the easiest way of doing that is this kind of hierarchy where you have densely connected recurrent networks all over the place. Information arises or ascends this system, creates a pattern at the highest level. Now here's the key. That pattern has to be stored in the lower modules at the time of acquisition. Okay, so the pattern is created up here. It's stored auto-associatively in the high level, and these levels down here may be storing attributes auto-associatively, but the connections from the bottom up to the hippocampus and from the hippocampus back down have to be modifiable so that the index can be associated with whatever is going on in whatever attribute is in the model at the time of acquisition and can later serve as a retrieval cue. So if there's some distribution at the input that creates a unique pattern, and later on there's a partial input that's sufficient to receive some of the information, if you get successful pattern completion in the highest level module, it's able indirectly to cause retrieval of patterns in the lower modules. So that's the fundamental idea. Now, we all know that the cortex is a highly modular structure. I'm not going to go into it. And module is a fuzzy concept, so we don't want to get bogged down on that. But I like this particular figure here, which shows the dolphin and mouse brain and the modular size in the somatosensory cortex, which these barrels are of the order 10,000 neurons, something like that. And that number seems to be preserved across a wide range of brain sizes. So again, this would be sort of a more modern cartoon of the index theory notion with the where now I've drawn these as Hopfield nets just to emphasize that they're storing information in the reciprocal connections using modifiable weights and re-emphasize the key features that these cortical modules themselves are content addressable memories, just like CA3. So, in summary, we have to postulate that there are actually several kinds of plasticity in this mechanism. There has to be plasticity from the ascending inputs, so this has to be because it has to be on the fly, it has to be done quickly, there isn't time for growth or rearrangement of connections. They have to use fast synaptic plasticity, LTP, LTD, and so forth. And you have to see that fast plasticity in the ascending inputs, in the descending inputs, intrahippocampal plasticity, and within the modules. And then there is what Jay and I call the slow learning system, which is the neocortex, and I'm going to get into a little bit of why Jay in particular came up with the idea that the cortex is slow learning. But I want to emphasize that that kind of slow learning is not the only thing that the cortex does. The cortex actually does fast learning as well, in the sense that it has to capture and store in the cortical modules information that's coming in on the fly, and it has to form these local attractors quickly. Okay, and I want to also emphasize that the return projections from the hippocampal formation to the cortex terminate primarily in the outer layers 2, 3 of the cortex, which, as Monahan and Cotman showed many years ago, when the NMD receptor was just being discovered, that is where you have the richest NMDA receptor concentration in the cortex. So that presumably reflects the plasticity of the back projections from the hippocampus back to the cortex, and possibly also attractor dynamics in the recurrent connections of the cortex itself, or the formation of attractors. Okay, so we have the standard model, but it's now on shaky ground. We have a fly in the ointment, which I attribute to my good friend Rob Sutherland starting to rock the boat increasingly hard around 2005, 2010, challenging a lot of the experimental evidence in particularly rodents for systems-level consolidation of episodic memory. Okay, and this is a paper well worth reading. Well, he rocked the boat so hard that eventually some people who were on board started abandoning ship, and nowadays there are very few people that would subscribe to the notion that episodic memories become independent of the hippocampus. That many kinds of memories, if they depended on the hippocampus to be formed in the first place, the memory itself continues to be dependent on the hippocampus. And then the question becomes, what is it that the cortex is doing and can still do? How do we explain the characteristics, for example, of HM, who had perfectly good semantic memory without a hippocampus, but was unable to form new semantic memory? And actually, I like to raise the point that the amount of episodic memory that the brain stores is trivial. Again, it's so small that if you were to model the brain, you might ask, you might neglect episodic memory altogether. Don't do it, but you might neglect it because the amount we actually retain is vanishingly small. What's left over, and what the brain is all about, is storing what psychologists call semantic memory or categorical memory, information about how the world works and how to predict the outcome of new events and our own actions. And again, this idea for me goes back to David Marr, who made models of the hippocampus and cortex in the early 70s, and about the neocortex, he said it may be regarded as a structure which classifies the information presented to it. One of the most important ways in which the nervous system eventually deals with sensor information is to form concepts with which to decompose and classify it. So now a lot of people have thought about these issues. I like this particular depiction because it raises an important point. There are things in the world, things about the world that are so regular and preserved across evolution that evolution was able to create the appropriate feature detectors and set up the brain such that simple developmental plasticity and so on can develop the most effective features at the lowest levels of the cortical hierarchy. But there are many, many things in the world that you wouldn't be able to predict by evolution, that change radically from generation to generation. If somebody years ago had showed me one of these tablet computers, I would have, been totally blind about, what its function was and so forth. So basically, all of these high-level features, actually evolution can predict some of them like faces and so on, but many of the high-level features need to be developed by the brain integrating the statistical properties of the world to make a valid statistical representation of the properties of the input and creating the most efficient feature detectors for it. So this pretty much is what is now called deep learning in the machine learning field, but the machine learning field didn't invent the concept, evolution did. So here is now the modern notion of what is involved when you take out the hippocampus. It leaves semantic memory relatively intact, but it disrupts all new episodic memory and all new category formation and semantic learning. Why? Because the episodes are the raw material of the semantic knowledge. They're the statistical samples of the world from which this categorical structure is going to be created. And that brings me to my one of my most fruitful collaborations that is going on still today with Jay McNaughton, who actually is responsible for most of the deep ideas in this paper. I added a little neurobiological plausibility, but following David Marr's input, he raised the idea, and which we explored in this paper, that the neocortex learns slowly to discover the structure in the ensemble of experiences. And at the time we were doing it, it was shortly after the connectionist revival. As some of you may know, that connectionism in artificial intelligence had a bad name for many years because of the lack of a way of teaching the synapses at the lower levels. That problem was solved, and there was an explosion of new conceptual ideas about how knowledge is represented in the brain. In our paper, we explored a variation of a model from Dave Rummelhart, in which we looked at a concept formation network. Basically, we had a bunch of items and a bunch of properties, and the idea was to teach the network with back propagation that when you gave a certain set of inputs to recall the proper outputs. And we looked at the representation in this hidden layer, which started off with random weights. And if you look over the course of learning, what you see is that the responses of the hidden layer become increasingly differentiated as learning progresses. Okay, it doesn't happen all at once, it happens in little steps. But what you see here is that the internal representation becomes, by EPIC 500, very categorically organized. You can tell the animals from the plants, and within categories you can tell differences between different exemplars, and that's captured in this sort of hierarchical clustering notion. So what's going on in this model is over many, many training trials, in which the whole corpus of input is repeated many times and small adjustments are made to the synaptic weights, the network sort of discovers the categorical structure of the input. And then we addressed the central problem of connectionist learning at the time, which was known as catastrophic interference or catastrophic forgetting in a paper by McCloskey and Cohen. And in our version, we trained up our network, and then we came along and said, oh, by the way, now that you've learned all these properties, we want you to learn about a penguin, which is a little bit not consistent with what you've learned, because a penguin is a bird that can move, swim, grow, has wings, but it can't fly. Okay, and so when the network was trained, having learned everything else, trained over and over and over again with presentations of a penguin property, it learned it very, very well, but at the expense of complete forgetting of almost everything else it knew. That's the catastrophic interference, because the weights all became directed to the penguin and the other knowledge was overwritten. If, however, the penguin was presented to the network, interleaved with recapitulations of the other stuff the network already knew, then the penguin found a way to gracefully insert itself into the statistical structure of the network. It learned a little more slowly, but there was very little problem at the output. So this led to the idea that in order to accommodate new knowledge into a semantic network, you have to train it with interleaved the new stuff with the old stuff. And this is now a central problem in machine learning. Okay, so just to illustrate, this is maybe what would be the output of a network that had learned about politicians, and then we teach it about dogs without interleaving the politicians, and this might be the output. I pulled this off the net. It's a beautiful picture. And then on the contrary, if we train the network interleaving dogs and politicians, then we might get it right. So the problem, how can we interleave multiple trials that occur only once? And to solve that problem, David Marr came up with the idea that actually part of the transfer between the hippocampus and the cortex occurs during sleep, a time when there's sensory input is excluded, and he actually predicted that the hippocampal recent memory is played out of the hippocampus during periods of sleep. And that led us to the trace reactivation theory, which simply says that while the brain's not busy processing external inputs, it's chewing its cud. It's spontaneously reactivating the indices of recent experience, which is retrieving representations of that synchronously throughout the cortex, and that leads to the gradual reorganization of horizontal connections within the cortex, most likely by rewiring, because there aren't enough of those horizontal connections to sustain all possible associations, but there's plenty enough to sustain the associations that have actually occurred in the animal's life if you get the right connections. So that is almost certainly going to be a rewiring mechanism, which was heresy when I was a graduate student. So this is the basic complementary learning systems theory idea that during sleep, recent memory indices are replayed. That causes the cortex to retrieve the recent episodes, but at the same time, or in the same epochs, the cortex has to be spontaneously retrieving stuff it already knew so that you can have an interleaving of the old and the new. And somehow, a miracle happens, and from that comes categorical knowledge, which is something that's, I think, not well understood yet. There are two fundamental problems with this idea. There's not enough time to reactivate everything. And the cortex no longer has access to the original data anyway. It only has the semanticized versions of it. So these are two fundamental computational problems that haven't been properly yet addressed in the connectionist field or in the artificial neural network field. And this is an area that Jay and I have recently started to look into, and I hope we'll have something to say about that soon. All right, index codes. What makes a good index code? First of all, as I've said before, it doesn't have to have any content, and probably you could just use random patterns. And indeed, random patterns are optimal in some sense because they're orthogonal by definition. They're as uncorrelated as they can be. Okay? The index code needs to be retrievable by auto-association, not only in the hippocampus, but also in the cortex. And that means that it must use sparse coding. You cannot make an effective auto-associative network using highly distributed coding. The index has to be broadcast widely to the neocortex, and this creates a severe biological problem, which I'm going to talk about. It needs to be stored within each cortical module along with the attributes of the particular experience. And that leads to the prediction that you might expect to find index cells also in the neocortex, most likely in layers 2, 3, where the hippocampal back projects to. And finally, the hippocampal index code needs to be spontaneously reactivated while the brain's offline, and it should cause reactivation of the neocortex. And I'm now going to skip over a bunch of slides because I knew I put too many slides in, and most of this is old stuff, and just tell you that we now know that that's true. I doubt there's anybody in this room who doesn't know a little bit about memory trace reactivation in sleep. It occurs both in the hippocampus and the neocortex. What we don't yet know, nobody has yet actually shown definitively that reactivation in the hippocampus really does drive reactivation in the cortex. There's a lot of correlation, but whether there's a cause and effect there, we don't know yet. And I have a student working on this issue. So why are hippocampal place cells perfect index cells? So this is now the link between the notion that the hippocampus is a spatial, whatever that is, analyzer, and that it plays a role as fundamental in generating semantic knowledge. And so let's just go through some of the attributes of hippocampal place cells. The first one is that we've suspected for many years that the dentate gyrus is a specialized little network whose job it is to push patterns apart, so-called pattern separation. This was David Marr's idea, again, in his paper on the cerebellar cortex. I stole it in the late 80s, and Lynn Nadell and I published a sort of a conceptual treatise on it in 1990, and then Alessandro Travis and Edmund Rolls did a very nice computational analysis of this. And I think all of these things generally support the idea that there's a very simple way to pattern separate on the fly, and that is to project the input into a higher dimensional space using a constant number of active cells, and that by just random statistics pushes the patterns apart. And David Marr had an extensive analysis of that, but I was drawn to the fact that there's maybe 100,000 entorhinal inputs to the hippocampus, and it's expanded tenfold at the dentate gyrus, and that just screamed that it had to be a pattern separating device. Okay, so pattern separation and randomization. Well, what is about a spatial code that is good for indexing? It actually gives an index code almost for free for almost everything we do. If you have a code that is uniquely linked to places in space, or better said, that changes systematically as we move through space, and if that code is sparse, orthogonal, then you have for free just about the best index code that you can imagine. And so in the mid-90s, it became increasingly clear that the hippocampus was really being driven by internal dynamics, as Yuri mentioned in his talk, that it's the internal dynamics that are allocating cells to places on the basis of how the animal moves around in space, not on the basis of what happens there. And the importance of that is that if two things that are very similar happen in two different places, you get for free different index codes for them. And now we spent the next 10, 15 years trying to demonstrate that the primary factor that's driving the allocation of place fields in the hippocampal formation was movement of the animal in space. And I'm not going to go through all the data, but I think there's an overwhelming case to be made that it's path integration that primarily, except in a few situations, drives where particular cells will be firing. Now, we don't understand yet how. There's a lot of conflicting models, and it's not an answered question yet, but it's unquestionable that movement of the animal in space is sufficient to allocate place fields, and that those place fields then become associatively linked to whatever happens at that place, so that that same cells will be retrieved when the animal revisits. And of course, Cliff Kentros showed early on that if you block NMDA receptors, you get perfectly good place cells in the hippocampus, but they never get bound to place permanently. So the animal comes back the next day and there's a new set of place fields. And then Carol and I showed that in old animals that have defective LTP, the same thing happens. You get what look like perfectly good place fields, but the animal can come back into the same environment and you get a totally different map. The nicest paper that addresses or supports the role or the random nature of the hippocampal code is this paper, which came out by Rich and Albert Lee's group in 2014, where they put animals on a very long track and ran and looked at the distribution of many, many place fields all over this 50-meter track. And then they did very nice statistics on the allocation of cells to locations, or said better, allocation of locations to a given cell. And what they showed is that it's a totally random process. It fits perfectly with the Poisson process, where the probability, given that I'm firing here, it has no bearing on whether it's going to fire 10 centimeters away or 100 centimeters away. It's a memoryless process. And actually, Drew Moore in my group showed actually that you can have place fields actually one on top of the other, showing double cycles of phase precession in some cases, and in other cases you have fields that are quite far apart, and there's no way to predict it. So now we come to the more modern era, and I show this slide just to illustrate the power of this new calcium imaging technology, which I'm getting more and more interested in, and to represent the fact that even in this sort of simplified spatial environment, we weren't by any means the first to show this, but that hippocampal cells provide a sparse and orthogonal representation of the virtual environment. So by sparse, I mean that at any given point along the track, very few cells are firing, so that the pattern here and the pattern a little 20 or 30 centimeters away is completely uncorrelated, and the entire environment is nicely tessellated or covered, is a better word, with place fields. My student Dun Mao showed that the characteristics of place fields measured with calcium imaging or with silicon probes were very similar in this apparatus. But there's a problem now for the use of a spatial code for indexing. And we all know what the problem is, right? You're sitting in the same place and you're trying to remember 5 different lectures. How do you discriminate events that happen at different places? Different events that happen at the same place. And for that, the hippocampus invented rate remapping, okay? So it was shown by Jill and Stefan Lutgeb in the Moser lab that the same cells had place fields in the same place, but that the firing of the cell could be driven up and down dramatically by what was happening at that place. And so that leads to this sort of idea here, if you think about a vector space, that 2 different places are different subsets of the hippocampal neurons or different subspaces of the overall vector space, but within those subspaces, the vector can be moved around by sensory input. And that provides the uniqueness that the indexing system requires. And then Zaneta Navratilova showed that, in fact, this rate remapping is a learned phenomenon that actually there had been a long-standing issue about why place cells were directional on a linear track and not directional in random foraging, and she showed that actually after the first run ever on a track like this, when the animal came back the second time, the fields were different from the first time, so it's a learned phenomenon. I'm going to skip through a lot of this stuff, well, just to make a point. Everything in the brain, everything that happens in the cortex and probably subcortical structures as well, influences the firing of hippocampal cells. Enough said. The hippocampus has access to almost everything that's going on in the brain. There's prospective and retrospective modulations of firing rate depending on what's just happened. That's stuff in working memory. What's going to happen, that's stuff in the planning system, sensory information, and so forth. And so I wanted to put this slide in here because Howard, of course, was one of my strongest intellectual contenders. For many years, we fought over whether it was a map of space or a memory map, and Howard was right all along. It's both. But if you actually do the math, spatial location accounts for most of the variance. And moreover, two cells which seem to respond to something in one, or to different things in one place, will respond totally different. Let me say this differently. If something happens in place one, some cells are modulated by it. If you now go to place two and give the same event, it's a totally different population of cells that's modulated by it, which is what you want for an index code. And it seems that that modulation information is coming in via the lateral entorhinal cortex. Another nice thing about the hippocampal system is that we've known for many years that there's a gradient of spatial scale as you go from the dorsal to the ventral hippocampus in the rat, or posterior to anterior in the human, and that gives some very nice sort of generalization properties. It's a way of making the indices not completely randomized and possibly endows some generalization or association of things that happen nearby in space or time. A recent paper from Ziv et al. With these nice mini endoscopes showed that actually the hippocampal place code isn't stable across days, it's changing from day to day. Okay? Well, you can ask for a better attribute for an indexing system, because if you want to have a different index code for the same event that happens on different days, you want to change your index code. So that's not at all inconsistent with this idea. And we saw in Edvard's nice talk at the beginning that there's also a longer time based variation in the firing of lateral entorhinal cells, which is also perfect if you want to generate distinct index codes for similar events at different times. The Schnitzer lab has also shown that the dendritic spines in the hippocampus turn over spontaneously with a time course of about 2 weeks. So that is probably just shuffling the deck in order to keep generating new patterns. And then we've known also for many years that the hippocampal index code seems to be reactivated. Recent memories, there have been nice talks about this today. Those cells that fire together during behavior fire together during sleep. And that firing, this is important for what I'm going to say in a minute, occurs during hippocampal sharp wave ripples. So the sharp wave ripple is the expression of memory reactivation, generally speaking, in the hippocampus. Now, it's not only memory reactivation. It, now we know does a lot of other things, but during offline processing, and it's reactivating recent indices. And we now know, and I'm going to skip over these slides, that memory is also reactivated robustly in the neocortex at an accelerated speed. But as I said, we don't yet know whether the hippocampal retrieval is causally related to the cortical retrieval. But we do know from Yuri's work and other groups that if you block hippocampal sharp wave ripples, you do impair memory consolidation, so that's comforting. I'm going to have to skip, well, just to say that memory is retrieved in the cortex in slow wave sleep during periods of robust up-down state transitions. These are the slow oscillations. And if you look in detail at memory reactivation, the strongest memory reactivation occurs during the periods when the up-down state fluctuations are the most intense. And more recently, we've been looking in more detail at the characteristics of the upstate. And to make a long story short, upstates, if you normalize time, are characterized by a period of high firing rate at the beginning and high firing rate at the end before it goes down. And most of the reactivation is occurring in this initial state here. And that leads to the hypothesis that actually what might be going on in the late part of the upstate is the magical interleaving retrieval of old memories that we're looking for. So that's currently hypothesis. I'm going to have to skip a couple of things here because I will run out of time, only to point to the very nice talk we had earlier in the symposium from Dr. Yang showing that synaptic spine turnover in motor cortex, at least, is critically dependent on slow wave sleep. REM sleep plays another interesting role, but most of the new spines are added during slow wave sleep. And it would be interesting to actually go and look at parts of the cortex that seem to be more dependent on the hippocampus and to manipulate the hippocampus. So now I want to get on to the biological constraint of how the index code is sent back to the cortex. And this has to do with the trade-off between economy and efficiency. Okay? That is to say, the ability to transmit a lot of information over a very small information channel, because Marr thought that the hippocampus actually sent synapses to every pyramidal cell in the cortex. Well, it just can't happen. Okay, so there's a bottleneck, and skip that. I think everybody here pretty much knows the difference between sparse and distributed coding. The mystery was that when we started to look at the sparsity of coding in the hippocampal formation, it turns out that the most sparse coding is in the dentate gyrus. CA3 is still pretty sparse, but it gets less sparse and less sparse until by the time you get to the subiculum where it's being read back to the cortex, it's almost a fully distributed pattern. Now, Marr emphasized that sparse coding was necessary to store information, but on the other hand, distributed coding is the most effective way to get the most number of patterns out over the smallest number of channels. And so that leads to the idea, and Carol and I showed single unit data for this back in 2000, that the subiculum is much more distributed coding scheme than the earlier stations. So just to go back to this diagram here of NMDA receptors, one thing that struck me back then was if you look in the subiculum, there actually aren't very many NMDA receptors, which suggests that actually the subiculum is not there to store data, but is there to do some kind of data compression and send out back to the cortex a distributed code, which is a much more effective way of sending information to the cortex. And so we come up with this model where the data are compressed into distributed form in the subiculum, but that would mean they have to be unpacked again in the cortex because they have to be stored there. Okay? And I'm going to skip a few slides and focus on this one. Now, this is voltage-sensitive dye recording in Majid Hajrani's lab. And most of you probably didn't see what happened there, but this is the right hemisphere of the mouse cortex. It's coming back again. Here's where the sharp wave ripple happens. So watch up here what happens in the cortex during the sharp wave ripple. Okay, you can see, let's do that one more time real quick, and I'll see if I can stop this in place. that the hottest zone is the default mode cortex, the places where the hippocampus projects primarily back to. Then there's subsequent activation of a lot of other areas, but this is the hot spot. And so Radboud has been looking at this in detail, and I just want to show this one slide which shows, the previous was an average. If, on the other hand, you look at individual trials, you see that some trials, the cortical activation precedes the sharp wave ripple, and some trials, the sharp wave, the activation follows the sharp wave ripple, and there's a continuum, if this is the ripple here, of cortical activations before and versus cortical activations afterwards. And if you look at the multi-unit activity in the hippocampus, you can see that when the cortex is preceding the sharp wave ripple, there's a buildup of multi-unit activity up to the ripple. And then when the cortical activation follows, there's a hippocampal activity after the ripple, and then there's states in between that. So here's some pure speculation. You have a recent memory, you have attractors in the cortex that is modular, a lot of little modules keeping index or keeping attributes stored in the cortex, and if they go off spontaneously, they can possibly trigger the retrieval of a pattern, an index in the hippocampus, which would generate a ripple, and that would result in the index going back to the cortex, giving broad arousal, which would be manifest as an upstate in the cortex. That's for recent memory. For consolidated memory, you might find that the activation of the same two modules by random chance in the cortex causes pattern completion in the cortex, which is an upstate, which then triggers a sharp wave ripple in the hippocampus. So there's probably this range of dynamics going on. This is, again, pure speculation, but that's what we're thinking now. So now the coup de grace is that my student Dunmau actually started to look for place cells in the superficial cortex, which was predicted by the notion that the cortex has to have index cells in the cortex that are sparse and distributed, or sparsely coded. I have 3 minutes, so I'm going to talk real fast. And what he found was that, at least in virtual reality, 50% of the layer 2-3 cells, if you use the right calcium indicator, have the properties of hippocampal place cells. And going very quickly, that place-specific firing disappears if you lesion the hippocampus. So this sparse coding of location in the superficial cortex depends on hippocampal output. There are a number of other similarities. The cortical place code is not stable over days, and it sharpens up with experience, only if you have a hippocampus. But now, we're looking all over the cortex, and we're finding pretty much the same thing in many areas of the cortex. Okay, we don't find it, surprisingly, very much in visual cortex or V2, but almost everywhere else that we can access so far, we're finding, and particularly in the frontal regions, a high density of place cell-like firing. And then, of course, you go back to the literature and you ask, well, are we the first? Well, no, we aren't, because Yuri, of course, showed this before we ever started looking at it, but he didn't actually. Maybe Yuri realized it, but it was buried in the paper that you can find sparse coding cells all over the cortex. So I'm going to leave you with this sort of hypothetical schema that we can think about the layer 2-3 of the cortex. Main role is actually to store the context or the index code in an auto-associative retrievable fashion, and that the decodable information in the cortex is in the deeper layers. And that's sort of the end of my talk. I got there, and that's where the field stands today, at least in my lab.